So we are starting the next presentation. Sorry. So this is one on immune evasion, and we'll try and work out and understand how the parasite is able to evade the immune response and the complexities involved in that. So I just moved to that slide. So the title here is Immune Evasion in Malaria. And as we discussed in the previous session, we have that the parasite has a very complex life cycle. You saw how it has these multiple stages, infects different kinds of cells, and then moves from the sporozoid stage to a liver stage to a blood stage, and there also you have the blood cell infection and the gametocytes formation. So a very complex life cycle. And if you are developing a vaccine, then the vaccine should be able to, or even if you don't have a vaccine, the immune response of the human should actually be able to address all these different stages, and one should be able to inhibit parasite growth or kill parasites at different stages in case they evade a particular step of the immune response. So the first complexity in malaria is that it has a complex life cycle. Then, depending on the stage of development in the human, in the mammalian host, it can be extracellular or it can be intracellular. So sporozoids are extracellular, but hepatocytic infections, liver infections, and the RBC infections are intracellular. The parasite exists in different forms and shapes, is what I just said, that in each of these stages, you have different morphology of the parasite, different kinds of proteins being expressed on the surface, and it expresses a different set of its 5,000 gene pool at a particular time. So some proteins are commonly expressed, more housekeeping proteins, but other proteins it modifies and expresses different protein on the surface. So the immune system has a completely different set of proteins to recognize at different stages. Now the parasite also has different localizations during development and infecting different cell types, is what I said, also sort of overlaps with what we said earlier. And therefore different innate and adaptive immune mechanisms have evolved in parasite control and inflammation. So if you have to develop a vaccine for malaria, and as you know, there is still no highly efficacious vaccine for malaria, even though people have been trying for so many years. There are 2 vaccines that have shown medium efficacy, but they are not up to the mark so far. So thus, for any vaccine development, it is important to know the protective immune mechanisms that one must induce for an efficacious vaccine to be developed. Now, this is again the thing that I was talking about earlier, the same figure, but here we are only looking at the human. So if you have to have immunity to the pre-erythrocytic stage in an infection, then antibodies that are generated during the immune response can affect this pre-erythrocytic stage in the pre-erythrocytic, so sporozoid and the liver stage in different ways. Now, antibodies can either bind to the sporozoid and inhibit the motility of the sporozoid in the dermis of the skin or in the liver. So you can have antibody that recognize surface proteins, and because they bind to the sporozoid, they can probably inhibit the motility of the sporozoid, which is extremely important for it to migrate through different cells and reach the liver. The antibodies can bind to the sporozoid and facilitate phagocytosis. So they can bind to the sporozoid, and then by means of that, they can actually enhance the phagocytic process by which phagocytes can engulf the sporozoid and express the antigens on MHCs. So you can have facilitation of phagocytosis by monocytes of phagocytosis in the spleen or the liver. This is one thing that the antibodies could do. The other thing the antibodies against the sporozoid stage could do is to block sporozoid invasion into the hepatocyte. So if they bind the sporozoids, and perhaps they can inhibit the invasion of the sporozoid into the hepatocyte by preventing the interaction between the sporozoid ligand and the interaction on the surface receptors of the hepatocyte, which are required for the invasion process to happen. The other thing they can do is inhibit sporozoid development inside the hepatocyte. So there are various ways in which antibodies can inhibit sporozoid motility, sporozoid invasion of the hepatocytes, and sporozoid survival in the liver stages. Now, the production of high levels of antibodies is dependent on the T cell help. You know that there are T helper cells in the immune response, and these CD4 positive T cells can recognize epitopes present on the sporozoid surface, and they can help boost the antibody production and immune response in a natural infection. Now, when the parasite is inside the hepatocytes, it can become the target of CD4 positive T cells, T helper cells, or CD8 positive T cells. And the hepatocytes express MHC class 1 and class 2. And these can, and the and parasite molecules can be loaded on MHC class 1 and class 2. So these are the antigen-derived epitopes that are be presented on class 1 and class 2. T cells can kill the parasite. They can either kill them directly as killer cells, or they can do so by the action of cytokines. And the cytokines such as interferon gamma. Interferon gamma also induces nitric oxide synthase production, and this increases the level of nitric oxide, which can also kill parasites in the liver. So there are various ways in which the immune response acts on the liver infection. Then there are innate immune mechanisms. And these innate mechanisms actually involve type 1 interferon pathway, and these are induced by the parasite infection and are active against late Schizons or against reinfection. This has recently been shown that these innate immune mechanisms can also act against reinfection. Now, how is there immune evasion by infected RBCs? So one way that infected RBCs evade the immune system is by attaching directly to non-infected RBCs to form a structure called rosettes. But this is a very, very important and interesting phenomenon. That you have an infected RBC. An infected RBC now has to evade the immune response, right? So there is one mechanism by which, as I remember, I described in my previous talk that there are the knobs on the surface of the infected RBCs, and these knob-like structures have this protein called PFE and P1. This is important for cytoadhesion. So the same protein, PFE and P1, can also interact with complement receptor, CR1, on the surface of uninfected RBCs. And this infected RBC, seen over here, surrounded by infected RBC, can form rosette-like structures. And these rosettes are actually, they are here and shown here in the endothelial, near the endothelial cells in the vasculature. So this is a way, because you have an infected RBC that's surrounded by normal RBCs, and it evades the immune response in this manner. Now, recent studies, a 2020 paper by me and team, and I've given the link here, so in case anybody of you is interested, you can actually go and see this paper, showed that rosette formation is important for evasion of the immune response. So what they did was that they incubated immune cells, and incubated phagocytes in this case, with clinical samples of malaria-infected blood cells. They took malaria-infected blood cells from infected individuals, and they studied the level of rosette formation. And they found that the rate of rosetting, right, the formation of rosettes increased by 10 to 40% in the presence of phagocytes. That is, if you had phagocytes, then you had a greater greater formation of rosettes. They suggested that the immune cell, that is the phagocytes, trigger the malaria-infected cells to form rosettes. So there is some correlation between the presence of phagocytes, which is immune cells, and formation of rosettes. They next tried to understand how these immune cells were stimulating rosette formation. So they extracted different components from the blood and analyzed it, and tried to see which of these was important for rosette formation. And when they used a step-by-step approach, which also included some genetic maneuvering, they identified a substance called human insulin growth factor binding protein. And this is called IGFBP7, insulin growth factor binding protein 7, that was required for formation of rosettes. So when they added this protein, this IGFBP7, to different malaria cultures, parasite species, they found that in the absence of immune cells, it can still stimulate the formation of rosettes. So therefore, this is important, and it can actually stimulate. And that's one way that the immune cells use to stimulate rosette formation. Now, how do these malaria parasites sense that there is IGFBP7? And so what is the trigger to start formation of rosettes? It was speculated that this must involve parasite-derived molecules, that you must have some molecules that are displayed on the surface of infected RBCs, and there is some interaction and ignition happening. So they altered RBCs, and they filtered out components of the blood. And by doing so, they identified a number of molecules required for rosette formation. And some of these were thought to be produced by immune cells, which is in response to malaria infection, such as the one that I described earlier, the insulin growth factor binding protein 7. And others are present in the blood with or without the presence of parasite. Some are irrespective of the parasite, but some are overexpressed in the presence of the parasite. Now they checked whether rosette formation triggered by this insulin growth factor binding protein 7 could prevent the malaria-infected cells from being swallowed up and destroyed by the immune cells. So does rosetting triggered by IGFBP7 actually prevent the parasite from being destroyed by the immune cells, that is being phagocytosed by the immune cells? And they did show that the presence of this the presence of IGFBP7 in the presence of this protein, rosette formation increased, but the number of infected red blood cells that were engulfed by the immune cells was reduced, right? So there was an effect. And this tells you that rosette formation has some role to play, and then that the presence of certain proteins enhances rosette formation, and there are signals that are that cross, that the parasite senses in the blood and can increase or decrease rosette formation, and this causes prevention of the or protection of the infected RBC from the immune response. Now, another phenomenon in the malaria parasite, so rosette formation then becomes an immune protective mechanism. Now, antigenic polymorphism. This is another way that the parasite, now, antigenic polymorphism is seen in all organisms, right? But let's look at it in the context of malaria. Now, the malaria parasite, organisms, is prone to mutations. As DNA replication happens, there are the multiple replication cycles, daughter cells are being formed, daughter parasites are being formed, and DNA is replicating. And in this replication process, it's a normal error of DNA polymerase mediated replication. So you have some mutations coming in. In the malaria parasite, there is 1 to 0.7 10 to the power minus 9 mutations per base. And that's the rate of the error of the DNA polymerase. But since the Plasmodium parasites have a 24 to 72 hour blood cycle, there is a high probability that mutation can occur and generate different parasite types. Some mutation somewhere. Some of these can be deleterious mutations. So in that case, the parasites will not be able to survive. Some of these can be neutral mutations. Some of these can be mutations that give it an advantage. Some of these can be neutral completely. So in the mosquito stages, the parasite undergoes sexual reproduction, as we saw, and gametocytes generate haploid sporozoid progenies. Now, recombination can also occur in the mosquito stages. So this increases the occurrence of gene polymorphisms. One way is mutation, the other way is recombination, and this also increases gene polymorphisms. So for some antigens, hundreds of haplotypes have been observed. There are so many mutations and SNPs found there that you can have many SNP combinations and indel combinations leading to a large number of haplotypes. And polymorphism in these coding sequences can due to be either point mutations, as I said, so single nucleotide polymorphisms, or insertions of certain small segments, or deletions of segments. So many Plasmodium antigens also have repeat elements, and this can vary in size and number, or have repetitive units. So a range of polymorphisms. So in most cases, such diversity is a result of immune pressure, since these mutations are often selected in regions which can be recognized by antibodies or T cells. So this is important to understand, that a lot of mutations are generated, but which of the mutations gets selected? And the selection in the infection process in malaria can be largely due to the immune pressure. So if you have mutations in epitopes and antigenic epitopes, then the mutations can be selected for, right? So if a particular mutation is not recognized by an antibody, a mutation in an epitope is not recognized by an antibody, then this epitope is able to evade the antibody response, and this can multiply. And then the parasite carrying this mutation can multiply. So therefore, the antibody response, the immune response, serves here as a selection pressure for selection of particular deviants or particular mutations or parasites. The mutations in B cell epitopes abolish the recognition of parasites by antibodies, and this may lead to selection of parasites with a different haplotype, as I just described. But this is very important for vaccine development. Since antibody-based vaccine formulations that target polymorphic epitopes, so you have polymorphic epitopes, multiple epitopes, polymorphic epitopes, which have variations, they can have limited efficacy, or they can also select for variant for vaccine-resistant parasites. So this is an important issue to be understood. Now, we give you an example. EMA1 protein, apical membrane antigen 1 protein, which is a highly polymorphic antigen. And this was one of the vaccine, one of the targets, molecules being looked at for vaccine development. Now, this is very polymorphic. So it is possible to envisage that immunogens, as I say here, that induce broadly inhibitory antibodies that recognize multiple variants may circumvent polymorphism. So if you have antibodies that recognize multiple variants and not specific for a particular variant, can have an effect. But if you have very specific antibodies specific for a particular epitope but not for a polymorphic epitope, then they will not be efficacious. So it is important to identify structurally conserved constraints in different variants and to try and understand how new vaccines can target these polymorphic antigens, because antigenic polymorphism is a way of immune evasion. Now, when we talk of antigenic variation, this is another aspect. One was antigenic polymorphism. This is antigenic variation. We mentioned, I mentioned in a previous talk, cytoadherence. So this is your infected parasitized RBC, and this is the membrane of the RBC. And here you have this knob-like structure I was talking about. Its primary constituent of which is the PFEMP1, parasite erythrocyte membrane protein 1. And this PFEMP1 and other proteins also, I'm not mentioning the names here, but you also have riffin and adhesine and sequestrin molecules on the membrane. So what happens here is that this PFEMP1 interacts with adhesion molecules on the surface of the host cell endothelium. And these can include CD36, ICAM1, VCAM1, PCAM1, etc. Okay, and thrombospondylus. So these interactions are very critical for the cytoadhesion phenomenon to happen. So now let's come to what we're trying to talk about in antigenic variation. Now, antigens are prone to antigenic variation, as I said earlier, are often on the surface of infected RBCs, and the primary player in this in P. falciparum is the VAR gene, and the VAR genes code for PFEMP1. But there are also Stivol gene family, there is a riffin gene family, and the sulfin gene family. There's also Sika-VAR gene family. And the Plasmodium interspersed repeats are also what are called the PIR genes. So the main one is the VAR genes, but you also have Stivols and riffins and sulfins and Sika-VAR. So now the VAR gene family is the largest. It's been shown to be the target of protective anti-blood stage antibodies. And the VAR genes code for PFEMP1. And as I said, these are highly polymorphic. They have different variable domains, and these domains are called DBL-like domains, DBL domains, Duffy binding-like domains. And these determine the binding specificities to different ligands on the surface of the endothelial cell. And as I showed you in the previous figure, these ligands can be thrombospondin, CD36, or ICAM, or PCAM, or chondroitin sulfate, E-selectin, various. And PFEMP1 proteins can also bind to RBC through the complement receptor to form rosettes. So, right, remember I talked about rosettes previously, and I said that PFEMP1 and CR1 interactions help in formation of the rosette, in which you have a parasitized RBC and surrounded by unparasitized normal RBCs. So, they are very important for adhesion and for rosette formation. Now, PFEMP1 is encoded by a VAR gene family, which are primarily located on telomeres of chromosomes in the malaria parasite. And it possesses roughly 60 VAR genes. And this expression of these VAR genes is extremely regulated. And only one PFEMP1 type, okay? So you have multiple VAR genes, but they're so regulated that only one of them, one PFEMP1 type, will be produced and displayed on the surface of an infected RBC. So at each cycle, the parasite can switch expression of the VAR gene, right? So if you have one cycle of infection, in the next cycle of infection in the RBC, the daughter parasites can switch the expression. So if one of these 60 was expressed in one line in previously, 2% of the parasites can switch the expression of VAR gene to another gene. And one parasite will express only one of them. So if you can change it at the rate of 2% in vitro, this generates a new clone with new antigenic and adhesive phenotype at each cycle. So if you have antibodies against one type, there might be 1 to 2% parasites in the next cycle, which will not be recognized by that. So this antigenic variation is, extremely important as an immune evasion strategy. Similarly, you have Vir proteins. Vir proteins are members of a superfamily of the PIR multigene family. They also mediate cytoadherence in P. vivax infected RBCs to endothelial cells, not in falciparum, but vivax. And this results in sequestration of the mature RBCs. There are about 350 Vir genes, and they're also highly polymorphic, like we saw in the VAR genes. Similarly, Stivol and riffin proteins that are involved in cytoadherence are also highly polymorphic. So all these cytoadherence molecules seem to have this high degree of variation. And because of the multi-gene families encoding them and their ability to switch, from one to the other during an infection, this is a big strategy for immune evasion. Now, some of these multi-gene family members have particular roles and may be involved in certain pathologies. For instance, if you look at the VAR gene 2 CSA, okay, this mediates a specific cytoadherence of infected RBCs to placenta. So if you have in pregnant women, you can the infected cells can go and adhere to the infected RBCs and coat and adhere to the placenta of the mother. And this is associated with placental pathologies of malaria. And this can cause stillbirth and fetus growth retardations. Now, if you have vaccination using VAR2 CSA-based immunogens, it is thus possible to induce inhibitory antibodies, prevent cytoadherence and placental substitution. So therefore it is important to understand what specific roles these variant antigens have in specific pathologies. And we must define the role of members of these multigene families in specific pathologies to tailor specific immune intervention, because these will have different roles in specific VAL genes can have different roles in different pathologies. The other strategy, or the other issue rather, is that there are redundant pathways of RBC invasion. What does this mean? So if one is worried about the fact that we want to inhibit the entry of merozoites into RBCs, that is, we want to inhibit the RBC infection process. So how are RBCs infected by merozoites? There's an initial contact. So the merozoite surface proteins come into contact with the RBC. There is reorientation of the merozoites, such as the apical complex is juxtaposed with the RBC membrane. And the microneum and rotary proteins are released, and there is invasion. And then after there is invasion, the merozoite enters and then gets converted into the ring stage, etc., etc. Now, how does this recognition of the merozoite surface proteins and the RBC proteins happen? As I said, it's a very complex and coordinated process. There is a process of orientation, there is a process of apical reorientation, there is a process of release of all these microneum and rotary proteins and dense granules at the apical end, and this invasion process has to be completed. Now, parasites can use multiple pathways. It was earlier thought there will be one conserved pathway of the invasion, but it's not so. Parasites can use multiple invasion pathways, and these involve at least 9 ligand receptor combinations to invade the RBC. So there are two parasite protein families in the host cell and the invasion process. One is the reticulocyte binding protein homologue, the RH, and the erythrocyte binding proteins, the EBNs. These are polymorphic again, okay, and their expression varies in parasite terms, right? So there's so much diversity. P. falciparum erythrocyte binding like proteins, EBLs, these polymorphisms can result in a change in the specificity for the receptor. So if you have a polymorphism in the EBL, the specificity it has for receptor recognition can alter. Or it can change in the switch to RH dependent invasion. So it can switch from an EBL dependent invasion to an RH dependent invasion, which is a different invasion pathway. So now's the crisis. If you inhibit EBL mediated invasion, there's a switch to RH mediated invasion. Now this can cause a lot of problems, because if you design vaccines against one, the other can take over as an invasion pathway. So they are able to overcome the inhibition that the immune system is providing through vaccine dependent targeting. Now, one thing that is encouraging in this is that pathway that involves RH5, there's another pathway that involves RH5 and its ligand Basogen CD147 on the RBC is required for all parasite clones and isolates tested so far. And it does not have so much polymorphism. So people are now working on this pathway to see if it's so conserved, then perhaps this is what we should use for targeting merozoite RBC invasion. Now, different from P. falciparum is P. vivax. P. vivax has, as you know, you must have heard, we have a Duffy antigen receptor on the RBCs, right? So humans have Duffy antigens, and the P. vivax parasite on its surface expresses what is called the Duffy binding protein. You can see it over here. So RBCs have a Duffy antigen receptor, and P. vivax expresses Duffy binding protein. If you design human monoclonal antibodies against the Duffy binding protein, you might be able to interfere this interaction, and therefore the infection of the reticulocytes. In this case, it's not the RBC. P. vivax infects reticulocytes. So reticulocyte infection can be inhibited. Now, contrary to P. falciparum, this was what was thought. It was thought that P. vivax has only one pathway to invade the reticulocyte, and this will only invade reticulocytes, which is the immature RBCs, and which is why people in West Africa or people with West African origins who lack the Duffy antigen were resistant to P. vivax. So if you don't have a Duffy antigen, then P-Vivax, Duffy binding protein will not be able to interact, and P-Vivax will not be able to infect. And this was an argument to the existence of only one invasion pathway for P-Vivax, that West African people who don't have the Duffy antigen receptor do not get infected with P-Vivax. But in recent years, there are several studies which have reported that P-Vivax infections also happen in Duffy negative individuals. So if you have an individual who lacks a Duffy antigen receptor, P-Vivax infections have been seen in such individuals. And so this suggests that the P-Vivax parasite merozoites can also use another pathway, and therefore they can invade reticulocytes from a pathway independent of the Duffy antigen receptor. Now, Duffy binding protein is a very leading sort of target for P-Vivax vaccines being developed. But this sort of such reports actually now make one think whether this will be such a good strategy. I mean, as good a strategy after all as one thought it would be, because the belief was that there is only one pathway for invasion, and now it turns out that you can have an alternative pathway. We don't know what the pathways might be, but that Duffy negative individuals can also get P-Vivax infection. So there must be another ways of infection of merozoites entering their reticulocytes. So therefore, this limits the efficacy of vaccines based on Duffy binding protein, and this might select for parasites that have been able to invade via the alternative pathway. And because if you do that, then the parasite or the automated pathway will still be able to invade, and will be able to select, actually selecting for them. So this is a problem that one faces for P-Vivax infections. So these are some of the complications that I just put across to you, because the malaria parasite vaccine, there are people who ask all the time that why haven't vaccines been developed for malaria? But it's not as simple as just taking a protein and expressing a recombinant and expecting to see an immune response that is completely protective. And immunoprophylaxis is not an easy thing for malaria. As it turns out, people have tried various things. They put multiple epitopes together. They put different, different strategies have been deployed. Different kind of antigens will be used from ANA1 to CSP to MSP and so on. The circumsporozoid protein-based vaccines seem to have done better than others. And as for Vivax, as I was telling you, Duffy binding antigen has been used as a strategy for countering Vivax. But work is in progress, and one hopes that with increased understanding of the immune responses as well as the immune evasion strategies, the kind of polymorphisms, the kind of antigenic variation, the kind of protein expression profiles that have now been worked out for stage-specific protein expression profiles, surface proteins that have been identified. I think with all that progress, perhaps one would have some better understanding of malaria pathology and immune evasion strategies, and these could be used for making vaccines. In the next presentation, which I will stop my this lecture now, and I will come back to the parasite again, and then we will talk of the other class, because that's a very interesting thing to look at in terms of parasite.